Namaste students. I'm Dr. Ajit from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth University, Jaipur. Today I'm going to discuss about the glyoxalate cycle. It is another process involving glycolytic enzymes and metabolites. The topic belongs to the subject fundamentals of microbiology and the BSc chemistry, second sem and Vidyapeeth second semester. Glyoxalate pathway is a series of metabolic reactions by which many different organisms utilize fats for the synthesis of the carbohydrates. It is an anabolic pathway occurring in plants and several microorganisms and fungi, but it is not found in the animals. Okay. Uh, It occurs in the glyoxysomes, the enzymes which are present in the glyoxysome cycles are common to TC cycle, and the enzymes include the lysozymes. The enzymes are known as the lysozymes, and one specific to mitochondria and the other is specific to the glyoxysomes. The difference is only that in the TC they are specific for the mitochondria, but in the glyoxysomes uh, they are specific for the glyoxysome. The glyoxylate cycle allows plant to use acetylcholine derived from the beta oxidation of fatty acids for carbohydrate synthesis. Okay. The glyoxylate cycle is regarded as an embolic variant of the citric acid pathway, and acetylcholine produced from the fatty acid oxidation condenses with the oxaloacetate to give citrate, which is then converted to the isocitrate. At this stage, isocitrate lies to succinate and glyoxylate. Another molecule of the acetylcholine is now utilized to combine with the glyoxylate to form malate synthase, and the malate so from enters the citric acid cycle. The glyoxylate cycle is a cyclic pathway that results in the conversion of two carbon fragments of acetyl forming four carbon compounds. Then succinate is converted to the oxaloacetate, and then glucose is involved in the reaction of gluconeogenesis. What are the reactions of the glyoxylate cycle? What are the different steps of the glyoxylate cycles? The glyoxylate cycle is a cycle which is related similar to the citric acid pathway. First, acetylcholine produced from the fatty acid oxidant will condenses to with oxaloacetate to give citrate. Okay. That is acetylcholine produced from the fatty acid oxidation. It will condenses to oxaloacetate to give citrate, and which is the citrate will be further converted into the isocitrate. First product is citrate, then isocitrate. At this step, isocitrate bypass the citric acid cycle, and it is cleaved by the isocitrate lies to succinate and glyoxylate. First, the acetylcholine condenses with oxaloacetate to give citrate. This citrate will be further converted into the isocitrate. Okay. Then isocitrate will bypass the citric acid cycle, and it is cleaved by the isocitrate lies to succinate and glyoxylate. Then another molecule of acetylcholine is now utilized to combine with the glyoxylate to form malate, and this reaction is catalyzed by the malate synthase, and the malate so form enters to the citric acid cycle. The glyoxalate cycle is cyclic pathway that results in the conversion of two carbon fragments of the acetyl bromine to four carbon compound succinate. That succinate is converted to the oxaloacetate and then to glucose involving the reaction to gluconeogenesis. So this is the main things of glyphosate cycle. What are the similarities of glyphosate cycle to the TCA cycle? The glyphosate cycle utilize the five of the eight enzymes associated with the tricarboxylic acid cycle. It is very similar to the TCA cycle. So it is there are eight associated enzymes like citrate synthase, aconitase, succinate dehydrogenase, fumarase, and malate dehydrogenase. These enzymes are similar, and the two cycles differ in that of the glyoxylate cycle. Isocitrate is converted into the glyoxylate and succinate, but the isocitrate lies instead of so alpha keto deuterate. The bypasses it will bypasses the decarboxylation steps that take place in the citric acid cycle, allow the simple carbon compound to get the synthesis of the macromolecules, including glucose. Now, what is the role of this glyoxylate cycle in the gluconeogenesis? From fatty acid to lipids are commonly used as an energy source of the vertebrates as a fatty acids are degraded so the beta oxidation into the acetate molecules. Okay. This acetate bound to the active thiol group of coenzyme A enters the citric acid cycle where it is fully oxidized to carbon dioxide, and this pathway allows cells to obtain energy from fat. To utilize acetate from fat for biosynthesis of the carbohydrate, the glyoxylate cycle, whose initial reactions are identical to the TCA cycles, are used. Okay. So the cell wall containing organisms such as the plants, fungi, bacteria require very large amount of the carbohydrates during growth for the biosynthesis complex structural polysaccharides such as cellulose, glucans, chitin. In these organisms, the absence of available carbohydrates, for example, in the microbial components of during seed germination in plants, the glycosylated cycle permits the synthesis of glucose from lipids via acetate generated in the fatty acid beta oxidation. So when the nutrients are not available and only fat is there, the gluconeogenesis pathway will take place, and the glyoxylate pathway will help the gluconeogenesis pathway to start. It will support the gluconeogenesis pathway. Okay, because some organisms do not have uh, glucose at a particular time. So what will they do? They will use the available fats which is present in them. Okay. This fat is available by the glyoxylate pathway. This glyoxylate pathway bypass the steps in the citric acid path cycle where carbon is lost in the form of the CO2. And the two initial steps of the glyoxylate cycle are identical to those in the citric acid pathway, acetates, citrate, isocitrate. In the next step, the catalyzed with the first glyoxylate cycle enzyme, isocitrate glides. Isocitrate glides it undergoes cleavage into the succinate and glyoxylate. The glyoxylate condenses with the acetylcholine. Okay, the glyoxylate condenses with the acetylcholine. Okay, which is catalyzed by the malate synthesis, and it will yield malate. What malate in the oxaloacetate can be converted into the phosphenol pyruvate, which is the product of the phosphenol pyruvate carboxygenase. The enzyme, the first enzyme is the gluconeogenesis. Okay, phosphenol pyruvate carboxylase is the first enzyme in the gluconeogenesis step gluconeogenesis pathway, which is the product of which is the net result of the glyoxalate cycle is therefore the production of glucose from the fatty acids. So it is helping in the first step of the gluconeogenesis. Okay. So this is the result of glyoxalate pathway, and it is also helpful in the another organism like in the plants. In plants, the glyoxalate cycle occurs in the specific peroxisomes, which are called the glyoxysomes, and this cycle allows seeds to use lipids as a source of the energy to form soup during the germination. If there is not an energy, the plants are utilizing lipids for energy. The seed cannot produce biomass reaching photosynthesis because of the lack of the organ to perform the function, and the lipid stores germinating seeds are used for the formation of the carbohydrates that fuel the growth and development of the organism. Okay. The glyoxylate cycle also can provide plants with another aspects of the metabolic diversity. This cycle allows plants to take acetate both as a carbon source and as a source of the energy. And acetate is converted into acetylcholine. This acetylcholine can proceed through the glyoxylate cycle, and some succinate is released during this cycle. And, and this glyoxylate pathway is also useful for the pathogenic fungi. The glyoxylate cycle may serve as an entirely different purpose in some species of pathogenic fungi. The levels of the main enzymes of the glyoxylate cycle, ICL and MS, are greatly increased upon contact with the human host. So in some vertebrates, it is also very helpful. How the glyoxylate cycle is inhibited due to the central role of the glyoxylate cycle in the metabolism of the pathogenic species, including the fungi and bacteria. Enzyme of the glyoxylate cycle are current inhibition targets for the treatment of disease. Most of reported inhibitors of the glyoxylate cycle target the first enzyme of the cycle, ICL. Inhibitors were reported for the candida albicans for potential use as antifungal agents. Different aspects are being used for the inhibition of the inhibition of the glyoxylate cycle. So, in this lecture, I discuss about some important aspects of the glyoxylate cycle. What are the different steps of the glyoxylate cycles, and how the glyoxylate cycles can be operated? What are the different importance of the glyoxylate cycles in gluconeogenesis, and what is the role of the glyoxylate cycle in the plants in the fungus? So, I hope the students will better understand all these things. So, my lecture is over here. If you have any query, you can ask me. So, thank you very much.